2001, A Space Odyssey, The Jetsons, The Moon Landing. The 1960s was a decade full of optimism towards the future of science and technology. This hopeful speculation wasn't just limited to popular culture, though. In 1965, Emile, Zuckerkandl and Linus published a paper titled Molecules as Documents of Evolutionary History. This paper discussed ways that genetic molecules, like DNA, RNA, or even proteins, could be used to investigate an organism's evolutionary past. So, were the theories and predictions this paper made ahead of its time, like 2001, or optimistically naive, like the Jetsons? Let's find out together, shall we? In the past, information like physical characteristics would be used to investigate an animal's evolutionary history and ancestors. The ideas around molecular phylogenetics presented in the 1965 paper were revolutionary and became the spark behind decades of research and the development of new methods of determining phylogenetic relationships. In their paper, Zuckerkandl and Pauling classified different molecules by how much information they could contain about the organism's evolutionary history. Molecules fell under three categories with decreasing importance to phylogenetics. The main hypothesis that the authors presented was this. They proposed that one species' semantic molecules, the genetic information like DNA, RNA, or polypeptide, could be compared to another species. Then the similarities between them could be used to determine how related they are on an evolutionary scale. Before the discovery of molecular phylogenetics, in the days of Charles Darwin, relatedness was determined using the difference in phenotypic traits alone. In their paper, they also proposed that synonymous mutations could also be useful information in determining evolutionary relatedness. A codon that contains a synonymous mutation between organisms is isosemantic to the other. Mutations can then occur on these isosemantic codons to change the amino acids that they code for. While not initially evoking change in amino acids, isosemantic substitutions can be important stepping stones between major mutations which affect protein function. Mutations that affect protein function can occur in multiple steps, meaning that these intermediate steps could be found in other organisms or species by comparing the nucleotide sequence of the allele in question. The existence of isosemantic substitutions thereby shows that the DNA sequence of an organism contains more evolutionary information than the polypeptide sequence. The paper doesn't detail how this information would be used, it only hints that the differences in semen tides could be quantified, but it laid the foundation for common practices of phylogeny that are still used today. After this information was published, methods of using semantic molecules for phylogenetics began to develop alongside technology. One of the first of these methods was published by Fitch and Margoliash in 1967. They used the number of mutational steps required to transition between two species' cytochrome C protein to quantify how related the two species are. This was the advent of what is now known as the distance-based method, one that transforms the number of differences in nucleotide sequence into a matrix. Another use of this method came in 1977, when researchers used the number of homologous sequences between Archaeal species' ribosomal RNA to generate an association coefficient. They used the association coefficient as a measure of the mutation distance and created a tree based on this. As technology developed, new methods joined the field of molecular phylogeny. The max and parsimony method became popular, which involves deeming the simplest possible phylogenetic tree to be the closest to the true evolution of the species involved. This method was exemplified in a 1982 project which used multiple proteins across 11 species to generate 39 trees out of over 34 million possibilities. The methods used in molecular phylogenetics today combine multiple methods, and due to the capacities of modern computers, thousands of species can be compared at once, including multiple genes, and complex algorithms are used to generate phylogenetic trees. Often, multiple techniques and methods of molecular phylogeny are used on the one dataset. In 2013, the complete classifications of over 1,400 bony fish species was published and then updated in 2018 to include 1,990 total species. This was done across 21 molecular markers using molecular and genomic data. Modern statistical approaches to phylogeny include specialized programs, often using hundreds or thousands of data points. Many of the predictions made by the authors haven't developed into fields of practice, but the things they suggested laid the groundwork for decades of research. While molecular phylogenetics isn't quite what Zuckerkandl and Pauling imagined in 1965, the techniques used today are able to accomplish their goal of using molecular analysis to unlock the secrets of an organism's history.